In this video, we will be discussing about the genetic disorders from the pathologic perspective. These notes are Dr. Devesh Mishra's notes from EDPMA learning. So let us start with genome. Genetics. Genetics means study of genes, right? And in our genome, 1.5% of human genome is coding, means it consists of exons, and only and 98.5% means the major part of human genome is non-coding, that is introns. What is the role of introns? So, that is also equally important. So the non-coding part consists of telomeres and centromeres, non-coding RNA, and jumping genes, right? Telomeres and centromeres are structural part of a chromosome. Non-coding RNA has different functions. Three types of non-coding RNA: microRNA, long non-coding RNA, and piRNA. Right. Jumping genes or transposons are functional in chromatin organization. Right. Jumping genes or transposon means these genes, they move continuously within the human genome. Now let us discuss about the non-coding RNAs. First is microRNA, denoted as small m small I RNA. It is 5% of human genome, and its function is silencing the other RNAs. For example, if miRNA suppresses the silences the tumor suppressor gene, the tumor can occur. If it suppresses the oncogene or proto-oncogene, so there will be no tumor. So it is functional in regulation of cell cycle, regulation of proliferation, when to check the proliferation and when not to check the proliferation. So gene can be knocked in, right? Gene can be knocked out and gene can be knocked down with the help of genetic engineering, which uses miRNA or microRNA. Now second type of non-coding RNA is LNC, long non-coding RNA. 200 to 500 nucleotides is the size. Its function is gene silencing or gene activation. Silencing occurs in the formation of bar body, that is silencing of 1X gene in a female, a 1X chromosome in a female. Second example of gene activation is to maintain the tertiary structure of a protein. So certain genes need to be activated to stabilize the structure of protein by synthesizing other proteins, uh, to make a tertiary three-dimensional structure of protein. So that is helpful in regulating chromatin organization and gene activity, right? So these are the two types of non-coding RNAs of human genome. Now let us discuss the genetic disorders, the main part of this video, the important part as far as exams are concerned. Genetic disorders can be divided into three categories. This is a very, very, very important classification. With the help of this classification only, we can mind map the things, that if something is asked, from where it is asked. So this classification is very important. I think there is no forgiveness if we forget this classification. So first is the single gene disorders, means disorders of one gene. Single gene disorders can be inherited in two ways. One is called classical inheritance. Mendelian inheritance is also known as Mendelian inheritance, which are we reading from last 10 years. So classical inheritance is one thing, and unclassical inheritance is another way in which these genes can be inherited. Mendelian inheritance is the classic inheritance. There are four mechanisms which can be autosomal recessive, autosomal dominant, X-linked recessive, or X-linked dominant. Right. Most common inborn error of metabolism occur by autosomal recessive mode of inheritance, and most common pattern of diseases occurs in autosomal dominant fashion. Right. Inborn errors of metabolism are inherited by autosomal recessive fashion, and otherwise most common pattern is autosomal dominant pattern of inheritance. Unclassical inheritance is one thing which is more difficult and more frequently asked. Four mechanisms of unclassical inheritance, that is trinucleotide repeats, mitochondrial inheritance, genomic imprinting, gonadal mosaicism. Trinucleotide repeats, mitochondrial inheritance, genomic imprinting, and gonadal mosaicism. So this is about single gene disorders: classical inheritance, unclassical inheritance. Now multiple gene disorders means multiple genes and environmental factors are involved in pathogenesis of a disease. Three diseases needs to be remembered: hypertension, coronary artery disease, and diabetes mellitus. These are polygenic multifactorial uh, pathologies uh, which are involved in pathogenesis of these three diseases. Though congenital heart disease, gout is also important, but three diseases, because when we read this diabetes mellitus, there is always written that it is multifactorial, environmental factors also play a role. So this is a complex polygenic inheritance mode of uh, method of inheritance, method of genetic disease, right? Now third kind of disorder that we often read is chromosomal disorders, means the whole of the chromosome is involved. Either there can be deletion, there can be duplication, there can be increase in the number of total chromosome, there can be decrease in the number of total chromosome, meiotic non-disjunction, mitotic non-disjunction. I mean, these are all the methods of pathology that are studied under the heading of chromosomal disorders. To cut short, there are two methods of chromosomal disorders. One is numerical disorders, means the number of chromosome has altered. Second is the structural disorders, means the number is same but structure of one chromosome or two chromosome is altered. Structural disorders can occur in following 4 or 5 ways. One is deletion. There can be deletion of a chromosome or a part of chromosome. There can be translocation. Translocation is very important as far as leukemias, lymphomas are concerned. For example, translocation 922, which is seen in CML, that is BCR ABL fusion gene and its functional protein. Third is the ring chromosome formation. Fourth is the isochromosome, and fifth is the inversion. Inversion is also a mode of genetic disorder in leukemias and lymphomas. So chromosomal disorders, there can be numerical, there can be structural disorders. Now these are the autosomal dominant and autosomal recessive uh, methods of inheritance. The Knudsen's two hit hypothesis, I think this needs to be remembered. It is classically seen in retinoblastoma. Means it tells us about the autosomal recessive pattern of inheritance. Means if one retinoblastoma gene, one RB gene is mutated, there is mutation M1, and other gene is normal. It is called as heterozygous uh, genome, right? Heterozygous genome is normal because the disease is autosomal recessive. Means if two genes are non-functional, only then the disease will occur. If one gene is non-functional, there will be no disease because one gene is normal and it is doing its function. If spontaneous mutation occurs after the birth of the individual, for example M2, there is M1, the initial mutation which was there. It can be before birth, it can be after birth, and there is another mutation in the another allele of the same cell, there will be loss of heterozygosity. Means the difference between the two will be lost, both will get mutated, and it can lead to formation of tumor, because the normal cell is now converted into a tumor cell because the pattern of inheritance or pattern of uh, inheritance is autosomal recessive. Right. So this is Knutson's two-hit hypothesis. So now let us discuss about these three disorders: Ehlers-Danlos, osteogenesis imperfecta, and Marfan. These are autosomal dominant disorders. These are the example of dominant negative effect. What do we mean by dominant negative effect? It means that if a gene is coding for 3-4 proteins, and out of these 3 or 4 proteins, one protein is non-functional because of mutation, it occurs in autosomal dominant pattern of inheritance only. Then the other 3 normal proteins will also not be able to function, which is seen in Marfan syndrome, Ehlers-Danlos syndrome, and osteogenesis imperfecta. Because these are the disorders of the proteins, structural proteins, for example, fibrin and collagen. These are very complex proteins, and they uh, have many, helices or many chains. And if one of those chains is affected, then the whole of the structure of the protein will be affected. So this forms an example of autosomal of a dominant negative effect, which is read in autosomal dominant pattern of inheritance. So these are three disorders. You can always pause the video and look at the uh, pathology and clinical features, and the genes and the proteins involved. You can pause the video and read these three disorders because these are important, right? Now next to be discussed is pedigree analysis. First of all, we must understand the pedigree symbols. Female is denoted by a circle, male is denoted by a square, right? Carriers are denoted by half shaded squares and circles. Deceased ones are denoted by fully shaded circles and squares. That is all I think that needs to be remembered in pedigree. Also, there are other symbols also. One is important is the carrier. Carrier means there is a dot inside the circle or dot inside the square, right? So let us discuss about the different patterns of inheritance with the help of pedigree. First is gonadal mosaicism. We must understand that this is unclassical pattern of inheritance. It is not Mendelian. Means the parents were normal. Only the children are affected. Means the mutation has occurred after mating or after fertilization, or at the level of gonads. Means the parents are not carrying the disease themselves, or they are not carrying the uh, defective gene in themselves, but at the level of gametes, at the level of zygote, or at the level of division after zygotes, there is some kind of mutation that has occurred in them. So that is gonadal mosaicism, is at the level of gonads. Examples are achondroplasia and osteogenesis imperfecta. Second is mitochondrial inheritance. It is also an example of non-classical or unclassical inheritance. It is not Mendelian, it is unclassical inheritance. Here we know that the mitochondrial DNA, the mitochondria is recruited from the female only. The male mitochondrial genes are destroyed, right? Sperm mitochondria undergo selective degradation. The only mitochondria which is passed on, mitochondrial DNA which is passed on to the next generation, comes from the mother. So the pattern of inheritance will be like ki mother is affected, then all the children will be affected. If the male is affected, right? If the male is affected, then and the female is normal, then the children will not be affected, because the male mitochondria will not be passed on to the next generation. Only female mitochondria will pass on to the next generation, not the male one, right? Example is, see, 4 5 examples we need to remember because they will be asked times and again. One is LH1, Leber's hereditary optic neuropathy. It is the prototype disease. Second is the MELAS, mitochondrial encephalopathy, lactic acidosis, and stroke-like features. It is most common. Second is the MERF. It is not written in there, but it is also ragged red fibers. Mitochondrial encephalopathy with ragged red fibers. Fourth is the NARP, neurogenic weakness and retinitis pigmentosa. Right? There are others also, but these 3 4 we must be able to remember. So these are two unclassical forms of inheritance. Let us study about the other unclassical forms of inheritance also. One is the genomic imprinting. It is a normal phenomena. Right? There is inactivation of one gene out of the two. Well, means one gene has two alleles, one from the mother, one from the father. One of them must be inactivated, and one of them must be active, uh, in order for the normal function of the cell to occur. But so how the other gene, the second one is inactivated? That is with the help of genomic imprinting. It is a part of epigenetics, right? Epigenetics means hereditary chemical modification of DNA or histones. How that chemical modification occurs? With the help of methylation or deacetylation, there will be reduced expression of that gene where methylation or deacetylation has occurred. Chromosome 15q is associated with genomic imprinting. Why we read chromosome 15 only? Because two very important genetic disorders are associated with chromosome 15q. One is Prader-Willi and one is Angelman. Normally, one gene has to be imprinted and other gene has to be activated, either paternal or maternal. If uh, if after genomic imprinting there is deletion, there is uniparental disomy, or there is abnormal imprinting of the normal gene, then disease will occur. Let me repeat it once again. One of the two alleles, either paternal or maternal, has undergone genetic imprinting, which is a normal phenomena. Now the second gene, right? Second gene, which is normal, it undergoes deletion, right? Or it undergoes uniparental disomy, means the two genes they come from one parent only, or there is abnormal genomic imprinting, then the disease will occur. Let us see how it occurs. For example, uh, maternal, for example, let us take the example of deletion. If maternal imprinting has occurred, which is normal, right? It is normal, but there is paternal deletion. If there is no paternal gene, there will be Prader-Willi syndrome, PW. Similarly, if there is paternal imprinting, which is normal, and there is maternal deletion, then there will be Angelman syndrome. Let us remember it by, if there is no maternal, there will be Angelman. If there is no paternal, there will be Prader-Willi. So deletion of paternal will be Prader-Willi, deletion of maternal will be Angelman, and other gene has already normally imprinted, normally inactivated, but the active one has been deleted in these two disorders. This is one mechanism, that is deletion. Second is uniparental disomy. What happens in Prader-Willi normally? Normally, the maternal gene, means mother, maternal gene is imprinted or inactivated, and paternal gene has to work normally, right? Paternal gene has to work normally, but what happened in here? There is no inheritance of paternal gene. There is duplication of maternal gene, which is already inactivated. So there will be two inactivated genes. Again, no paternal means P. No paternal means P, that is Prader-Willi, right? So this is example of uniparental disomy of maternal gene. If there is uniparental disomy of paternal gene, means the two genes come from father only. No mother means M, man syndrome, Angelman syndrome, right? So these need to be remembered and revised times and again in order for them to in order for you to remember them. Now let us discuss the clinical features of Prader-Willi syndrome. Prader-Willi syndrome, remember we also study this syndrome in obesity. Because of why obesity occurs? Because of increased release of ghrelin hormone, which is orexigenic hormone, which increases appetite. Normally it is secreted from the antrum of stomach. There will be mental retardation and hypogonadism also. So this will be read in endocrinology in great detail. Two pathologies, hypogonadism and obesity, and it is a genetic disorder. So mental retardation also. These three things, obesity, mental retardation, hypogonadism, is the feature of Prader-Willi syndrome. Angelman is very easy to remember. Angel means something which is good, which is smiling, which is laughing. But this is Angelman syndrome. Here the laughter is very inappropriate. Inappropriate laughter and ataxia, ataxic movements, will be clinical features of Angelman syndrome, also known as happy puppet syndrome. Angelman means M means no mother. No mother means no mother, no gene from the maternal side. Maternal genes are deleted, or the father's inactivated genes have doubled. Right? This is the Angelman syndrome. Other examples of uniparental disomy are Russell-Silver syndrome and Beckwith-Wiedemann syndrome, which is associated with Williams tumor. Right? So this is the third unclassical method of inheritance, that is genomic imprinting. Fourth unclassical mechanism of inheritance is trinucleotide repeats. Normally trinucleotides get repeated in our genome, but when these repeats occur more than what is required, means 6 to 55 times is normal, more than 55 times is abnormal, up to 200 we can tolerate. It is called as pre-mutation. It will be the patient will be a carrier, there will be no symptoms. But as soon as the repeats go more than 200, there will be overt disease. The symptoms will start appearing. Number of repeats is proportional to severity of disorder. Increase in number of repeats during oogenesis and spermatogenesis occurs, right? The time when repeats occur is oogenesis, spermatogenesis, or gametogenesis. We can also call it gametogenesis, right? This is trinucleotide repeats. Examples: Huntington's, spinocerebellar ataxia, myotonic dystrophy, Fragile X syndrome, Friedreich ataxia. It is written oogenesis में or spermatogenesis में, but it's okay. Don't stress over this too much. Huntington disease and Fragile X, these are the examples of trinucleotide repeats that need to be remembered. Again, Huntington and Fragile X, trinucleotide repeats. Right, these are the four unclassical forms of inheritance. Right, we have studied trinucleotide repeats, mitochondrial inheritance, genomic imprinting. इसमें हमने पढ़ा Prader-Willi और Angelman. गोनेडल मोजाइकिज्म में भी कुछ पढ़ा था. माइटोकॉन्ड्रियल इनहेरिटेंस में लाज़मेर्फ. ट्राइन्यूक्लियोटाइड रिपीट में फ्रेजाइल एक्स and spinocerebellar ataxia, and one more thing. What was it? Okay, we'll read. Now, classical inheritance: autosomal recessive dominant, X-linked recessive, X-linked dominant. These can be studied with the help of pedigree, but I am not going into the details. You can always pause the video and you can read on this page that there is X-linked recessive right? X-linked dominant, autosomal recessive, and autosomal dominant. 4 patterns. There is Y-linked disorders also given. So pause the video, read this page. You will read the classical inheritance, Mendelian inheritance. Right. Now let us jump on to the chromosomal disorders. There can be structural, there can be numerical. Structural means the structure of a chromosome is affected. There can be deletion, ring chromosome formation, isochromosome formation. There can be, translocation, inversion also. In numerical disorders, there will be, decrease or increase in the number of mutations. If there is one autosomal chromosome, one autosome is in excess, it is called as trisomy. And if it is one chromosome in deficiency, it is called as monosomy. So if one chromosome 16 is extra, it is trisomy 16. The patient will die in utero. If one chromosome 21 is extra, it will be called as trisomy 21, which is Down syndrome. It is most common chromosomal abnormality in live birth. That is Down syndrome, trisomy 21. Now there are 3 methods of investigation. One is karyotyping. Karyotyping means study of the chromosomes, right? Second is FISH, fluorescent in situ hybridization. It is there for the detection of known gene loci. If you know the gene where the defect can be, you can always put some corresponding nucleotides so that they attach to that particular locus and give a certain fluorescence under the detector. So you know the gene, you put that nucleotide pair or nucleotide chain. So for the purpose of detection of known gene loci, you can use FISH. But what if there is point mutation? There is no gene but a point mutation, a single base pair in the gene is affected. For that purpose we have PCR, right? So PCR, FISH, and karyotyping, we must be able to remember what are the methods and what are they used for. Karyotyping is basically the morphology. We study the morphology of chromosomes. Now numerical abnormality: deletion, trisomy of autosomes, and deletion or trisomy of sex chromosomes can be there. First of all, deletion. If there is deletion of chromosome 5p, there will be Cry-Duchet syndrome. Autosomal cytogenetic disorder. If there will be trisomy 21, it will be Down. Trisomy 13 means Patau, and trisomy 18 means Edwards syndrome. Associated with sex chromosome, if there is one X chromosome extra, it will be Klinefelter. If there is one X or one Y chromosome deficient, there will be Turner syndrome. Down syndrome is the most common cytogenetic abnormality which is detected at birth. It is trisomy 21, pathogenesis 3 mechanisms: meiotic non-disjunction, Robertsonian translocation, and mosaicism. Most common method of inheritance is meiotic non-disjunction, which is very important question which was asked in NEET SS 2023. Okay, so that is why it is very important to remember the methods of mutations or methods of chromosomal abnormalities. Clinical features of Down syndrome is flat facial profile, mental retardation, right? Most common genetic cause of mental retardation is Down syndrome. Oblique palpebral fissures or mongoloid slant, not stent, slant. Mongoloid slant. Brush field spots, whitish spots on the iris, and prominent epicanthal folds low set ears, scrotum tongue, like large protruding tongue with wrinkling. Congenital heart disease will be there. Endocardial cushion defect is most common. Then there will be VST, then there will be AST. Hands are short, broad. Fifth finger is curvature. Sorry. Fifth finger's curvature will be there. It will be not straight. It will be like a curve. The little finger will be in a curve. Single palmar crease, also known as simian crease. Other features are duodenal atresia, megacolon, intestinal obstruction, hypotonia, and wide gap between first and second toe. Most common leukemia in Down syndrome is ALL more than AML. Most common AML is M7. Most common leukemia occurs in less than 3 years old. See, Alzheimer's disease occurs because of deposition of amyloid precursor protein inside the neurons or outside the neurons in the form of uh, neurofibrillary tangles and tau proteins. APP, amyloid precursor protein. It is coded by chromosome 21. What happens in Down syndrome? There is extra chromosome 21. There will be more APP protein, more misfolding, more deposition, more chances of Alzheimer's disease. So universally, in case of Down syndrome, beyond 40 years, there will be universal affection by Alzheimer's disease. Very important, needs to be remembered. These are serum markers. Turner syndrome, Klinefelter syndrome. You can always pause the video and have a look at this and remember this. Right. Now, diagnosis of genetic disorders. It can be by PCR. It can be by FISH. It can be by microarray. 3 mechanisms: PCR, FISH, microarray. Otherwise, for the detection of defects in DNA, we use southern blot. For RNA, it is northern blot. For protein, it is western blot. Right? It is asked a multiple number of times, so it is a part of cramming. So I think we must remember this. As we have already read, FISH is for detection of known gene loci. Microarray is for comparative genomic hybridization for detection of unknown gene loci. Microarray. For example, we use microarray in gene expression profiling and in molecular classification of cancer. In gene expression profiling, we can understand whether the tumor expresses estrogen receptors or progesterone receptors or HER2 new receptors. Molecular classification of cancer is also used in CA breast. So this is the use of microarray. So that is the major part of human genetics as far as pathologic aspect is concerned. The medicine or the therapeutic part of genetics we will be discussing in some other video. So that is all about this thing. Thank you so much for your patient listening.